Viruses are acellular infectious agents that exist in the extracellular state as virions, composed of a nucleic acid genome encased in a protective protein shell called the capsid. The genome itself displays extraordinary diversity. It may consist of DNA or RNA, be single or double stranded, and can take a linear or circular arrangement. Now, RNA viruses are further classified as positive sense, which can function directly as messenger RNA, or negative sense, which must first be transcribed into a complementary strand by RNA replicase. Structural organization further distinguishes viruses. Now, the capsid may display helical symmetry, as in the tobacco mosaic virus, or icosahedral symmetry, as in adenoviruses. Now, some viruses exhibit highly complex morphologies, such as bacteriophage T4, with its icosahedral head and helical tail apparatus. In many cases, viruses also possess envelopes derived from host cell membranes into which viral glycoproteins are inserted. Now, these glycoproteins mediate host cell recognition and entry, but also serve as major antigenic targets for the immune system, making them critical both for pathogenesis and the vaccine design. Viruses often carry essential enzymes packaged within the virion. For instance, bacteriophage may contain lysozyme like proteins to degrade bacterial peptidoglycan, facilitating DNA injection. RNA viruses frequently encode RNA dependent RNA polymerases for replication of their genome, while retroviruses carry reverse transcriptase, integrase, and protease to facilitate conversion of RNA into DNA and stable integration into host genomes. Now, these enzymatic features allow viruses to overcome host restrictions and initiate productive infection, even in environments where host enzymes alone would be insufficient. The interplay of genome type, structural symmetry, and enzymatic content determines how each virus replicates, interacts with its host, and causes diseases. Now, importantly, these features also influence strategies for antiviral development, since inhibitors can be designed to target unique viral enzymes, structural proteins, or envelope glycoproteins. Quick recall fill in. Number one, the extracellular form of a virus is called a blank. Number two, a single stranded RNA genome that has the same sequence as messenger RNA is called blank sense. Number three, blank symmetry describes viruses that are rod shaped. Multiple choice questions. Number one, which of the following is not a viral genome type? Number two, icosahedral symmetry is best described as. Number three, which enzyme is carried by retroviruses? Mix and match challenge. Application case. A researcher isolates a new virus with an RNA genome. When tested, the RNA is immediately translatable by ribosomes. What type of genome is this, and how does it differ from negative sense RNA viruses in replication? The viral life cycle is a highly ordered and subsequential process that begins when a virion recognizes and attaches to a specific receptor on the surface of a susceptible host cell. This attachment step is crucial as it determines host range and tissue specificity. Following attachment, penetration occurs, during which the viral nucleic acid, or in the case of some animal viruses, the entire virion, is delivered into the host cell. Uncoding may occur simultaneously or shortly after penetration, releasing the viral genome into the cytoplasm or nucleus. Depending on the virus, once inside, the virus enters the synthesis stage where it hijacks the host machinery to replicate its genome and direct the production of structural proteins, enzymes, and regulatory molecules. The assembly stage follows, in which newly synthesized viral components spontaneously self organize into complete virions. Finally, virions are released either through host cell lysis or, as in most bacteriophages, or through budding, as in many enveloped animal viruses. The success of these stages depends on the permissive host cell that provides the required enzymatic and structural machinery for replication. The one step growth curve provides a framework to visualize these processes experimentally. After viral entry, the eclipse phase is observed, in which no infectious particles are detectable because virions have disassembled and the viral genome is being expressed and replicated. This phase highlights the hidden intracellular replication that is invisible to assays measuring extracellular virions. The maturation stage follows, marked by the assembly of capsids, packaging of genes, and formation of infectious particles within the host. The release phase explains the dramatic rise seen in viral count during the growth curve experiments. In bacteriophages, host cell lysis liberates hundreds of virions at once, defining the burst size, a key parameter in viral kinetics. In animal viruses, release may be more gradual through budding, which spares the host cell and allows persistent infections. The mode of release influences not only the pathogenesis but also immune system recognition as budding viruses cloak themselves in host membranes. Quick recall fill in. Number one, the stage where the viral genome is replicated and proteins are synthesized is blank. Number two, the eclipse phase occurs when blank. Number three, a host that supports full viral replication is called blank. Multiple choice questions. Number one, the eclipse phase is characterized by. Number two, which of the following correctly represents the order of viral replication? Number three, in a one-step growth curve, the rise period corresponds to. Mix and match challenge. Application case. A student is culturing E. coli and infects the culture with a bacteriophage. When measuring viral counts over time, they notice a long period where no virions are detected, followed by a sudden sharp increase. What does this suggest about the viral growth curve, and which stages explain the sudden increase? Bacteriophage T4 has a complex genome and intricate replication cycle. Unlike similar viruses, T4 encodes its own DNA replication machinery, including DNA polymerase, primase, helicases, and accessory proteins, which together form a phage specific replisome. Its genome exhibits circular permutation and terminal redundancy, allowing complete packaging into the plasmids even when cut at different sites within concatemeric DNA. Packaging is driven by a powerful ATP dependent motor, which forces the DNA into preformed proheads. The temporal regulation of early, middle, and late gene expression ensures orderly replication, host cell takeover, and structural assembly. Lambda phage has adopted an alternative replication strategy known as lysogeny. In this state, the viral genome integrates into the host chromosome as a prophage or persists extra chromosomally as a plasmid and replicates in synchrony with the host without producing new virions. Lysogeny is maintained by repressor proteins such as cytoplasmic incompatibility 1, which blocks transcription of lytic genes. This provides the virus with stability during conditions unfavorable for rapid replication while still allowing for eventual activation of the lytic cycle. The decision between lysis and lysogeny is a finely balanced regulatory process. Controller of repressor's operator or CRO protein promotes lysis by inhibiting C1 or cytoplasmic incompatibility 1 expression, while C2 and C3 proteins stabilize lysogeny by protecting C1 from degradation. Environmental stressors such as UV irradiation or DNA damage can trigger prophage induction, excising the viral genome from the host chromosome and initiating lytic replication. Beyond replication, temperate phages contribute to bacterial evolution through transduction, the process of packaging host DNA into viral particles and transferring it to new hosts. This horizontal gene transfer is a major drive of genetic diversity and can spread traits such as antibiotic resistance or virulent factors. Quick recall fill in. Number 1. The lambda phage genome integrates into the host genome with the help of blank. Number 2. The protein that represses transcription of lambda phage lytic genes is blank. Number 3. Phage T4 encodes its own blank for genome replication. Multiple choice questions. Number 1. Which protein determines entry into the lysogenic pathway in lambda phage? Number 2. A prophage refers to. Number 3. Phage T4 modifies its DNA with blank to resist host restriction enzymes. Mix and match challenge. Application case. A culture of E. coli infected with lambda phage remains viable over many generations without visible lysis. However, after UV irradiation, the culture undergoes mast cell death due to phage release. What viral mechanism explains this observation? Animal viruses differ fundamentally from bacterial phages in both entry and replication strategies. Whereas bacterial phages inject nucleic acid into bacterial hosts, animal viruses typically deliver the entire virion into the host cell. Entry occurs either through direct membrane fusion, common in envelope viruses such as HIV, or endocytosis, where the host engulfs the virion in a vesicle. Following entry, uncoding releases the genome into the cytoplasm or nucleus. This step is crucial for determining the site of replication, as DNA viruses often replicate in the nucleus, while many RNA viruses replicate in the cytoplasm. Animal virus infections can follow several distinct pathways. In a virulent infection, the host cell is lysed as progeny virions are released. In a latent infection, the viral genome persists in the host without producing infectious particles until reactivated by stimuli such as stress. A persistent infection occurs when virions are continuously released by budding, allowing the host cell to survive, but producing long term infections, as seen in hepatitis viruses. Finally, a transformation occurs when viral genes alter host regulatory pathways, converting normal cells into tumor cells. This highlights the oncogenic potential of viruses such as human papilloma virus and Epstein Barr virus, sometimes activating oncogenes or disrupting tumor suppressor genes. Retroviruses, including HIV, utilize a unique replication strategy that requires the enzyme reverse transcriptase. After entry, their positive single strand RNA genome is reverse transcribed into DNA, which is then integrated into the host genome by integrase. This proviral DNA is stably inherited by daughter cells, ensuring persistence. Viral proteases subsequently process polyproteins during maturation, enabling infectious virions assembly. Because of this integration step, retroviruses can induce permanent genetic changes, sometimes activating oncogenes or disrupting tumor suppressor genes. Antiviral therapies frequently target viral enzymes such as reverse transcriptase, protease, and integrase, while vaccines aim to elicit immune response against viral glycoproteins. Quick recall fill in. Number 1. The process where animal viruses enter by engulfment is called blank. Number 2. The viral enzyme that integrates retroviral DNA into the host genome is blank. Number 3. A viral infection that results in tumor formation is called blank. Multiple choice questions. Number 1. Which infection outcome involves continuous virion release without host lysis? Number 2. Retroviruses differ from other RNA viruses because. Number 3. Which of the following is not a retroviral enzyme? Mix and match challenge. Application case. A patient's biopsy reveals that normal epithelial cells have undergone uncontrolled growth after infection with an oncogenic virus. Analysis shows viral DNA integrated into the host genome near a proto-oncogene. Which viral infection outcome explains this transformation, and which enzyme was necessary for integration?